Hello everyone. In this video, there are 50 MCQs on nucleic acids. The fundamental unit of genetic information is known as DNA, protein, gene, RNA. The correct answer is gene. Nucleic acids are polymers of nucleotides, nucleosides, DNA, RNA. The answer is nucleotides. Chromatin is composed of DNA only, histone proteins only, DNA and RNA, DNA and histone proteins. The answer is DNA and histone proteins. What is the overall charge on a DNA molecule under physiological conditions? Positive, negative, neutral, depends on the sequence. The answer is negative. DNA fingerprinting recognizes the difference in nitrogenous bases, sugar molecules, phosphate groups, short tandem repeats. The answer is short tandem repeats. How do nucleotides link together to form long chains in nucleic acids like DNA and RNA? Hydrogen bonds, peptide bonds, phosphodiester bonds, glycosidic bonds. The answer is phosphodiester bonds. Which of the following is not a component of nucleotide? Phosphate group, nitrogenous base, fatty acid, pentose sugar. The answer is fatty acid. Which of the following correctly describes the bond that forms between 3' hydroxyl group of one nucleotide and the 5' phosphate group of the next nucleotide in nucleic acids? Hydrogen bond, phosphodiester bond, peptide bond, glycosidic bond. The answer is phosphodiester bond. The bond that holds the two DNA strands together. Ionic bond, covalent bond, hydrogen bond, peptide bond. The answer is hydrogen bond. DNA controls protein synthesis through the mediation of which molecule? Ribosomes, enzymes, RNA, lipids. The answer is RNA. Which pyrimidine base is found in DNA but not in RNA? Cytosine, uracil, thymine, adenine. The answer is thymine. The strand of DNA that is encoded with the genes is called template strand, coding strand, complementary strand, antisense strand. The answer is coding strand. Which of the following correctly describes the components of a nucleotide found in DNA? Ribose sugar, nitrogenous base, phosphate group, deoxyribose sugar, purine or pyrimidine base, phosphate group. Deoxyribose sugar, amino acid and phosphate group. Ribose sugar, pyrimidine base only and phosphate group. The correct answer is deoxyribose sugar, purine or pyrimidine base and phosphate group. Ribose and deoxyribose differ in their structure at which carbon? 1' carbon, 2' carbon, 3' carbon, 5' carbon. The answer is 2' carbon. Why is the guanine cytosine base pair more stable than adenine thymine base pair in DNA? G and C are purines. G and C form ionic bonds. G and C form 3 hydrogen bonds while A and T form 2. G and C are larger molecules than A and T. The answer is G and C. They form 3 hydrogen bonds while A and T form only 2 hydrogen bonds. The melting temperature of a DNA duplex increases with higher adenine thymine content, lower pH, increased guanine cytosine content, presence of uracil. Answer is increased guanine cytosine content. The double helix structure of DNA was proposed by which scientist? Charles Darwin and Alfred Wallace, Watson and Crick, Gregor Mendel and Thomas Morgan, Franklin and Wilkins. The answer is Watson and Crick. A, B, and Z DNA are respectively left, right, and left respectively, right, right, and left respectively, left, left, and right respectively, right, left, and right respectively. The answer is right, right, and left respectively. A DNA is right handed, whereas B DNA is also right handed, whereas Z DNA is left handed DNA. According to the base pairing rules in DNA, which of the following pairs are correctly matched? Adenine cytosine, guanine thymine, adenine thymine, guanine cytosine, cytosine thymine. The answer is adenine thymine, guanine cytosine. Which nucleotide sequence is found at the 3' end of the acceptor arm of tRNA, where amino acid is attached? GUA, CCA, AUG, UAG. The answer is CCA. What is the primary function of the anticodon arm of tRNA? Bind the amino acid, catalyze the formation of peptide bonds, recognize and pair with the complementary codon on mRNA, stabilize the ribosome during translation. The answer is recognize and pair with the complementary codon on mRNA. In a DNA molecule where adenine makes up 30%, what is the total percentage of guanine and cytosine? 30%, 40%, 50%, 70%. Answer is 40%. If A is 30%, then T is equal to 30%. So that 40% is for G plus C. According to Chargaff's rule, in a DNA molecule, A is equal to C and G is equal to T. A is equal to G and C is equal to T. A is equal to T and G is equal to C. A is equal to U and G is equal to T. The answer is A is equal to T and G is equal to C. If the percentage of cytosine in a DNA sample is 22%, what is the percentage of guanine? 11%, 22%, 28%, 44%. Answer is 22%. G is equal to C. So, if C is equal to 22%, G must also be 22%. How many base pairs are present in each turn of the B form of the DNA helix? 9, 10, 11, 12. The answer is 10. The right-handed double helix of DNA is stabilized primarily by ionic bonds between phosphates, hydrophobic interactions between the base pairs, van der Waals forces between sugar phosphate backbone, hydrogen bonds between complementary base pairs. Answer is hydrogen bonds between complementary base pairs. The backbone of nucleic acid structure is constructed by which type of bond? Peptide bond, glycosidic bond, phosphodiester bridges, all of them. Answer is phosphodiester bridges. Which of the following coenzymes is a nucleotide? FAD, NAD+, coenzyme A, all of them. The answer is all of them. NAD plus and FAD are primarily involved in which type of biochemical reactions? Hydrolysis, redox reactions, phosphorylation, group transfer reactions. Answer is redox reactions. The nucleotide that serves as an intermediate for biosynthetic reaction. UDP glucose, CDP acylglycerol, S adenosyl methionine, all of them. Answer is all of them. If the DNA has the nitrogenous base sequence ATTGCC, what will be the corresponding mRNA sequence? TAACGG, UAACGG, ATTGCC, CGGTAA. Answer is UAACGG. During transcription, mRNA is synthesized from the DNA template strand by base pairing. So, for DNA ATTGCC, the corresponding mRNA sequence is UAACGG. What is the function of CCA sequence at the 3' end of tRNA? It stabilizes the tRNA structure by forming hydrogen bonds with the anticodon loop. It facilitates the binding of the ribosome to the tRNA during translation. It is the site where an amino acid is covalently attached. It helps in the recognition of the tRNA by the amino acyl tRNA synthetase. Answer is it is the site where an amino acid is covalently attached. Under physiological conditions, the DNA structure is predominantly in which form? A form, B form, Z form, triple helix. Answer is B form. Which scientist discovered that in DNA the amount of adenine equals thymine and the amount of guanine equals cytosine? James Watson, Erwin Chargaff, Rosalind Franklin, Francis Crick. Answer is Erwin Chargaff. zDNA differs from bDNA in that it is right-handed and has major grooves. It contains uracil instead of thymine. It is left-handed and zigzag in structure. It is found only in prokaryotes. The answer is it is left-handed and zigzag in structure. Which of the following nucleic acid techniques is best suited to quantify gene expression levels? Western blot, Southern blot, Northern blot, QRT-PCR. Answer is QRT-PCR. Which of the following statements about RNA is false? RNA is single-stranded. RNA is involved in protein synthesis. RNA contains a thymine instead of uracil. RNA has ribose sugar. Answer is RNA contains a thymine instead of uracil. Which strains of Streptococcus pneumoniae did Griffith use in his experiment? The S strain, R strain, both S and R strains, neither S nor R. Answer is both S and R strains. What is the significance of Griffith's experiment? It proved DNA is the genetic material. It discovered the process of transformation. It showed that proteins carry genetic information. It explained the structure of DNA. Answer is it discovered the process of transformation. In Griffith's experiment, what was the result when live non-virulent Streptococcus pneumoniae bacteria were mixed with heat-killed virulent bacteria? The non-virulent bacteria remained non-virulent. The non-virulent bacteria became virulent. The non-virulent bacteria died. No transformation occurred. The answer is non-virulent bacteria became virulent. What was the primary discovery from Griffith's experiment? DNA is the hereditary material. Bacteria can undergo transformation. RNA is responsible for genetic traits. Viruses cause bacterial infections. Answer is bacteria can undergo transformation. What was found with the experiments of Avery, MacLeod, and McCarthy? Proteins are the genetic material. DNA is the genetic material. RNA carries genetic information. Transformation does not occur in bacteria. The answer is DNA is the genetic material. Why did the mice survive when injected with only the heat killed S strain in the experiment? The bacteria became dormant, the bacteria were completely destroyed, the capsule of the S strain was removed, the R strain neutralized the S strain. Answer: the bacteria were completely destroyed. Avery, MacLeod, and McCarthy used which enzyme to prove that the DNA is the transforming principle? DNase, ligase, RNase, protease. Answer is DNase. Which organism did Avery, MacLeod, and McCarthy study in their experiment? Mice, viruses, bacteria, yeast. Answer is bacteria, Streptococcus pneumoniae. What was the primary objective of the Hershey Chase experiment in understanding genetic material? To prove proteins are the genetic material, to confirm that the DNA is the genetic material, to discover the structure of DNA, to demonstrate bacterial transformation. Answer is to confirm that the DNA is the genetic material. Which organism did Hershey use as a model in his experiment to study the nature of genetic material? Mice, bacteria, viruses, yeast. Answer is viruses. Which radioactive isotope did Hershey Chase use to specifically label the DNA of the virus in their experiment? Carbon 14, phosphorus 32, sulfur 34, nitrogen 15. Answer is phosphorus 32. What was the most important conclusion drawn from the Hershey Chase experiment? DNA enters the bacterial cell and directs viral replication. Proteins enter the bacterial cell during infection. DNA remains outside the bacterial cell. Both DNA and protein enter the bacterial cell. Answer is DNA enters the bacterial cell and directs viral replication. What was the purpose of using sulfur 35 in the Hershey Chase experiment? Sulfur is present in DNA. Sulfur is used for bacterial growth. Sulfur is radioactive and enters the bacterial cell. Sulfur is present in proteins, not in DNA. Answer is sulfur is present in proteins, not in DNA. Thank you.